Good morning, children. Today, in the sixth chapter, Molecular Basis of Inheritance, we are going to learn about Lac Operon. Francois Jacob and Jacques Monod in 1961 were the first ones to propose the concept of a transcriptionally regulated system where a polycistronic structural gene is regulated by a common promoter and regulatory genes. Such an arrangement is referred to as an operon. There are different examples of operon, like Lac Operon, Lac for lactose, Trip Operon for tryptophan, Era for arabinose, His for histidine, and valine, that is valine operon. Now, let us study about the structure of operon. It consists of various genes, like number 1 is the regulatory gene, that is the I gene, which codes for repressor. It codes for repressor of the Lac Operon. The term I does not refer to an inducer, rather it is derived from the word inhibitor. Then it consists of 3 structural genes. And the 3 structural genes are number 1 or A is the Z gene. Z gene codes for beta galactosidase that helps in the hydrolysis of disaccharide into its monomeric unit, that is lactose into galactose and glucose. The second structural gene is the Y gene. Y gene codes for permease that increases the permeability of the cell to beta galactosides. And the third one is A gene. A gene codes for a transacetylase. Okay, now let us study about lactose regulates switching on and off of the operon. Lactose is known to be the inducer and the substrate for the enzyme beta galactosidase. If lactose is provided as the carbon source in the growth medium, in the absence of the preferred carbon source such as glucose, the lactose is transported into the cells by the action of enzyme permease. The lactose then induces the operon. The I gene synthesizes the repressor of the operon. repressor binds to the operator region of the operon, preventing RNA polymerase from transcribing the operon, and is inactivated by interaction with the inducer such as lactose or allolactose. Now that the repressor is inactivated, RNA polymerase is allowed access to the promoter and transcription proceeds. In this figure, you can see how the lactose regulates switching on and off of the operon. This is the diagram of an operon where you can find the promoter region. Promoter is there. Operator, this purple color is the operator region, and these three blue color structures are nothing but the structural genes, that is the Z gene, Y gene and A gene. As I have already explained you, Z gene codes for beta galactosidase that helps in the hydrolysis of disaccharide into the monomeric units, whereas the Y gene codes for permease that increases the permeability of the cell to beta galactosides, and the A gene codes for transacetylase. Now, as I told you that lactose regulates the switching on and off of the operon, lactone is known as the inducer and the substrate for the enzyme beta galactosidase that is being coded by the Z gene. If lactose is provided as the medium, here you can see if lactose is present in the medium, then the lactose will be taken up, it will be transported into the cells by the action of the enzyme permease, and then the lactose induces the operon. Okay, now the I gene, okay, here the I gene codes for, or it syntheses the repressor of the operon. When the repressor is produced, the repressor binds to the operator region. The purple color region is the operator. So the repressor binds to the operator. As a result, the RNA polymerase cannot bind to the, it cannot bind, it cannot activate, okay, it cannot get activated, and there will be no interaction with the mRNA, and there will be no transcription of the genes. There will be no transcription of the structural genes. Okay. Now, if the repressor is inactivated, how the repressor will be inactivated? The repressor will be inactivated if in the medium the lactose sugar is present, because the lactose will get binded, it will bind to the repressor, and as a result the repressor will get inactivated. When the repressor gets inactivated, the RNA polymerase can access to the promoter, and the transcription of Z, Y and A genes will take place. Now, regulation of Lac Operon by repressor is referred to as negative regulation. Lac Operon can work under the control of positive regulation also. Children, here you can see this is the diagram. This is the diagram of an operon. Okay, where you can find the promoter regions. This is promoter. This is operator region. These are the structural genes. Okay, and this is the enzyme RNA polymerase. Now, the the gene I, it codes or it produces the mRNA that produces the repressor. Now, this repressor binds with the operator region if there is no lactose in the medium. Okay, and if the repressor binds to the operator region, there will be no access of RNA polymerase on the structural genes Z, Y and A, and as a result the gene Z, Y and A cannot produce different enzymes. that is enzymes beta-galactosidase, permease and trans-acetylase respectively. Okay, but if in the medium lactose is present, okay, lactose will be taken up by the cells and this lactose in turn will bind to the repressor. As a result, the structure of the repressor will change. Okay, and when the structure of the repressor changes, or you can say the repressor gets inactivated and it cannot bind to the operator region. When the repressor cannot bind to the operator region, the RNA polymerase can access to the promoter and transcription proceeds. Okay, so when lactose is absent, I gene regulates and produces repressor mRNA in the absence of lactose, which blocks transcription. The repressor protein binds to the operator region of the operon and as a result prevents RNA polymerase from binding to the operon. The operon will be switched off in this situation. When lactose is present, lactose acts as an inducer here and binds to the repressor. Thus, it forms an inactive repressor. The repressor fails to bind to the operator region. The RNA polymerase binds to the operator and transcribes Lac mRNA. That is, Lac mRNA is known to be polycistronic, which produces all three enzymes beta-galactosidase, permease and trans-acetylase. And in this situation, operon will be switched on. So children, that is all about the operon and Lac operon in detail. I hope you have understood this topic. In our next video, we will be learning about Human Genome Project. Thank you.